Welcome to the dkNET Introductory Webinar. I am Marianne Martone. I'm a professor at the University of California, San Diego, a professor emerita. I recently retired, but I am also with Hypothesis and SciCrunch. And I have Dr. Ko-Wei Lin with me, who is going to be monitoring the meeting. So thank you for taking the time out of your schedule to learn a bit more about dkNET. The way that the presentation will be structured is we'll first go over a general introduction to dkNET, a little bit about the structure and content, how dkNET works. We're then going to run through a couple of use cases, and we've decided to focus the use cases in this case on funding issues, because as we will say later, that is one of our most popular indexes. And we also recently presented at the ENDO conference, the Endocrine Society, and by far and away the most common questions about resources were about funding. And then we'll also talk a little bit about how dkNET can help with the NIH guidelines for rigor and reproducibility. And we'll talk a little bit about the dkNET NERSA challenge. My intention is to do live demos. I've been in this business long enough to know that live demos are always risky, so I have some backup slides just in case there are network problems. But again, if you're having any trouble hearing or please have any questions at all, please enter them into the chat and Ko-Wei will let me know about them. So let's start, first of all, with an overview of dkNET. dkNET stands for the NIDDK Information Network. It is a community resource and information portal that was set up by NIDDK for basic and clinical investigators to essentially provide the community with a means to communicate about, inform, and also discover diverse research and clinical resources that are available to you. Resources in this case means data, materials, tools, funding opportunities, also things that are available in the literature. And as we'll go through in a minute, the information about these research resources, of course, you can go to Google and you can search the web, but a lot of the information is found in these very large databases that have been set up either by the NCBI, but in many cases by a lot of individual centers. And these databases are typically what we call the hidden web. That is, they are not easily searchable. The contents of these databases are not easily searchable by a search engine. So we really focus our attention in dkNET on being able to provide tools and easy ways for researchers to search inside of these databases so that they can find the resources that they need. As it says in our title, we are also, however, an information network, and so we have various alert services, Twitter, and other things and forums for people in the community to keep in touch and abreast of opportunities that are available or just informational items that are available across the various NIDDK-funded resources. And as we will encourage you at the end, if you listening have any information that you'd like to share with the community, we'd like you to let us know so that we can pass it on. So we're first going to go through the ways that you search in dkNET and some of the search tools that are available. But the second part of the presentation will also focus on the fact that dkNET is a key infrastructure and infrastructure provider, and provides you tools and infrastructure for identifying and tracking research resources. This has become a very important issue with the new emphasis on rigor and reproducibility, and in fact NIH has now issued guidelines, and we'll show you how dkNET can help with this. So first of all, by way of introduction, one of dkNET's prime functions is to essentially make people aware of the vast number of resources that have been created for them, the vast number of these databases. And you can see over on the right a listing of all of the databases that dkNET accesses. There's currently over 200, and we search about 1 billion records from these databases. On the left, you see a word cloud that kind of gives you some idea of all of the different things that are available. And down in the right little graph, you can see that there's a wide variety of resource types that are represented in dkNET in its various databases. So what does dkNET do? dkNET essentially makes them easy to search and browse. If you use any of these databases or you go online, you realize that every single one of them has a different user interface. They have different types of information. The way they display information is different. And suppose the thing that you want is found in 4 or 5 of these. Well, having to go to 4 or 5 different databases, even presuming that you know that they exist, is actually fairly difficult because you have to learn how to navigate each one of these. So dkNET essentially makes it very easy for you to search and browse through these databases. And I want to emphasize, and I will bring this up again, dkNET does not replace these databases, right? These things are extraordinarily valuable and extraordinarily rich. And if these particular databases are important to your research, it is well worth the time getting to learn about that specific database and use some of the tools that are provided for you. But again, many researchers are not aware that these databases exist. And if your information is scattered across multiple ones, we make it very easy, just like Google doesn't replace the web pages that it indexes, but it provides an easy way for you to search across them. So that's what this slide represents. So I want to talk a little bit about the structure of dkNET. We're constantly trying to improve this, but it's actually a fairly complicated thing to try to provide access to all of these hundreds of different databases and not overwhelm people with the amount of information that's available. So the way that we do this through dkNET is we've essentially divided up dkNET into 3 different indexes or sections, if you will. The first one, which we'll spend the bulk of time on today, are the community resources. These are specialized NIDDK relevant resources. We'll show a list of them in just a minute. They come from NIDDK funded centers and also represent general research resources like antibodies and cell lines that are of interest to almost every community. The second index is called More Resources, and it's got that little icon on the top because this set of resources, dkNET is built on the SciCrunch platform. You saw from my introductory slide that there's actually now a company that's been formed to help with the sustainability of this. But SciCrunch is the initial aggregator of all that information, and it does that for different communities, not just NIDDK, but also neuroscience and spinal cord injury and some of the other ones. And the idea of SciCrunch is that as it aggregates this information across these communities, it makes it available to everybody. But it's kind of like drinking from a fire hose. And so we wanted to make sure that we separated and highlighted those things that were uniquely valuable to NIDDK from this larger set of databases that are available to everybody. And finally, since most scientists spend most of their time in the literature, we also provide an index into PubMed. As we'll show very briefly, because we can control the interface to the literature, we can add features and things that aren't available through PubMed, but make it easy and interesting to browse the literature. So essentially, when you use dkNET to do a search, you search simultaneously across all 3 of these. And if dkNET sometimes work a little slowly, the reason why is it has to make a dynamic query into all of these databases and return those results to you. So here's the list before we do a live demo of how to use dkNET. We wanted to just provide some information on the types of resources that we have available. So you can see on the left, these are the NIDDK funded resource centers. We have things like NERSA, the Nuclear Receptor Signaling Atlas. The Beta Cell Biology Consortium data is available through dkNET. That resource has been sunsetted. And one of the important functions of dkNET is that we can keep the data and the valuable data that was generated by this particular project and still make that available through dkNET, even though the original website is no longer available. As new dkNET or NIDDK centers come online, such as HERN, we have the Human Islet Research Network, I believe that stands for, and I know that John is on the webinar. We, again, work with them not only to make their data available, but as we start to work across these communities, we can start to standardize how information is presented so that it's not so disruptive to people to go across these different research resources. But as I also mentioned, the community resources are supplemented from some of the large databases that have been provided for the general biomedical community. These include things like plasmids, clinicaltrials.gov, the antibody registry, and search across all of the model organisms databases. So this just gives you a list of what's currently in community resources. So I'm going to do a dangerous thing right now, and I'm going to, I hope, switch to the live demo coming to the center screen of dkNET. And the first thing I'm going to do is just do a general search. So if I'm coming to the search page of dkNET and I don't know what's in it, I'm just going to enter my term, in this case, type 1 diabetes. And you'll notice that it provides a list of terms underneath. This is not like Google, where it's representing searches that other people have made. Rather, these are terms that we have in our vocabulary services that we can use to expand the terms that you actually search for under a given concept. So here again, if it ends up being too slow, I won't, I'll switch over to slides, but that wasn't too bad. So if you go to this little box here that provides some insight into what we're actually searching, you see that when we pick from the autocomplete, instead of just searching for type 1 diabetes, we're searching for all of the synonyms and variants. And that's because even though there's a lot of push for people to use standard terms, I think you can all imagine that have been of limited success. And so rather than forcing different sources to use a terminology, we rather gather up and make these terms available for search. So that's what the expansion is. So when you come here, you've basically been placed into community resources. You can see that up here. Again, the search has been executed across these two different indexes, but we're going to focus our attention here. And you can see a list of resources that are available. They look like Google search results, but actually these are records from individual databases. And how do I know what I'm seeing? Well, if you go over to the left menu, you see first at the top that there are a series of options. So this essentially gives you access to some of the features that are available from dkNET, including make sure that you see only new records or the most recent records. But I want to focus our attention down here, where you can see that dkNET has given you a set of categories that represent the different types of things that you're seeing here. So for example, since we did an all search, it's showing you how many materials we have, how many funding related materials we have, and data and organisms. But if there's a plus sign, it'll expand it out. And you can see that of those 135 resources, there's antibodies, there's some vector, there doesn't look to be any vectors or primers, but there are, for example, 82 cell lines. The other thing that you'll notice is in addition to these categories, below that, we always list the source of the databases from which we are pulling records. So you can see that the records that came from this particular search come from all these different sources. So at any time, you can switch back and forth between the category and then the list of sources that provide data or resources within that category. So for example, if I was interested in cell lines, I could click on cell lines here. It brings me again to a list of records. And if I scroll down here, I see that all of these records, in fact, come from one source, which is Cellosaurus. And you'll notice here that, again, these things are presented as individual snippets. This makes it very easy for you to scroll through and look at these individual records. But if you're like me, many scientists like to look at databases not as a series of records, but as a table. And so there's a little table icon here that actually lets you select it, and then it will switch to a tabular view, which shows you all of these records now as a table. So here you can see down here the table of all the results. You see they come from type 1 diabetes. And up here, it gives you a little explanation of what Cellosaurus is. So even though there's only one source, the reason why we picked this source is, in fact, it has dozens of individual providers that all have been aggregated. So essentially, you can search across many, many different databases with an easy query. So that's basically, again, the way that dkNET search works. Once you get into some of the searches, you can see that you can expand columns. Also, at any point, if you want to download the results as a spreadsheet so that you can keep track of them, dkNET lets you do that. There are advanced features like Save Search and alerting services. We're not going to go through them today because, again, we find that most people, myself included, when I come to a new database, I tend to use the simple features, and it takes me a while to use some of the more advanced features. But these are all available. We do, in fact, maintain on the dkNET site a lot of help and tutorials if you ever get confused. Also, we provide search tips to help you narrow down your search. So I said we weren't going to spend a whole lot of time on the more resources and the literature because, again, I think those are topics for another day. But just to let people know a little bit about what's available, let me scroll through these slides. The more resources, essentially, again, we have a new interface that we've introduced that lets you use these categories to narrow down. So it asks you, what are you looking for? Are you looking for physical resources like antibodies? Are you looking for funding or general information? Gives you some subcategories, and then provides a list of resources that are available. So the more resources is interesting because we've selected a subset of resource types that are uniquely interesting to the NIDDK community based on the centers. But if you come to more resources, you find interesting things like databases that come from neuroscience that may have brain activation foci, for example, on type 1 diabetes, microarray experiments, proteome exchange, comparative toxicogenomic databases, all different kinds of things. So it's worth spending some time on that. And as I mentioned briefly, we also search the literature. Of course, you can go to PubMed or Google Scholar for this, but dkNET tries to add some additional features that make it interesting. One of the things that we do is we provide altmetric data. These are alternative ways of viewing at the impact of an article. So for example, if there are news stories on it, or people are blogging about it, or people tweet about it, it lets you know that these other sources of information are available. So just to very quickly review, what essentially we did with in searching dkNET is we entered in a broad term, type 1 diabetes. We then went into the community resources, and we used the filters and categories and facets that were available for the databases that were returned to be able to narrow down our search to the things that we were looking for. And this just provides this in a schematic view. We spent some time in community resources. We selected materials. Materials told us that cell lines were available. We selected cell lines, and it took us to Cellosaurus. So the reason that we put this slide in is that we're all used to searching search engines like Google. And in fact, when you search databases and structured information, you have to use a different strategy than you use when you're searching web pages. And so we like to remind people that the best way to start with dkNET is a general term, and then use the tools that we give you for refining and modifying your search, rather than starting with a search that has too many terms. And I don't want to go into the reason why, but it is the nature of databases that essentially means that the more effective strategy is to start general and then refine your search. Okay. So I thought again that since we had so much interest in using dkNET for funding, that we would do a specific use case, specifically looking at some of the resources that dkNET has available for funding. So again, we had a booth at the Endocrine Society. We got a lot of people coming by, and there was typically a question like this. I'm a postdoc interested in pursuing metabolomic research. How can I find information on funding? This wasn't an actual thing because, of course, we don't share confidences, but this was a general type of use case that was given to us. So we're now going to go through that again, only this time we're going to focus on specifically the resources we have available for funding. So I'm going to go back to dkNET. I always like to start at the beginning, although you can enter a search from anywhere. So how would I go about answering that query? Well, the first thing I would do is I would type in metabolomics. I see there is a term, but also note down here that you can write from the beginning, pre-select what you would like to see. So in this case, there's something called funding, and that's what I'm looking for. So I'm going to go ahead and do the search. So again, we're seeing the community resources tab, the list of options that you have available. We're going to scroll on down. And here we see that the subcategories that are available for funding actually fall into two categories. One is opportunities, and the other is funded grants. So we're first going to go to opportunities, and then we're going to look a little bit at the funded grants, because that's a really rich source of information, the NIH Reporter. So again, down below, it lists all the sources that are in this general query. We see we have something called grants.gov, which is a database maintained by the federal government that has all federal agency opportunities listed. The NIH Reporter, which we just mentioned, but also a specialized source that actually was created by us at dkNET called Pilot Funding News, and we'll go into that in just a minute. Natural thing for me to start here, since I was looking for funding opportunities, is just to select funding opportunities. And again, I see that there are two databases. There's grants.gov and dkNET pilot funding. I'd like to explore these 45 results here. So I'm going to come over and go into table view. And here's table view. You can see that this works like any spreadsheet. So you can filter by post date, you can filter by closing date, you can look at what agencies and things are available. But one of the problems that we've had with dkNET is that it has so much rich information. It's very hard to kind of get a sense of get overviews of what it is that dkNET is trying to show you. So one of the things that we've recently added is a word cloud feature. So whenever you see this little cloud icon up here and you click on it, it takes all the results from that particular column and it plots them relative to frequency. It takes the top 200. So in this case, it's telling you all the funding agencies that are available and how sort of frequent that is in the response. So not surprisingly, the major funder of metabolomics research is the National Institute of Health. That screams out at you. But I thought it was very interesting that when I looked at it, you see that actually NASA also funds it, and the National Science Foundation funds it. And if you dig into that a little bit more, you see that NASA has a space biology program where they actually fund metabolomic research. And so this is a very, interesting way to start to explore not the things that you know to be true, but also perhaps some things that you don't. I mentioned before that we also had a special database called Pilot. And up here in the right, there is a navigation. You can also use your browser back bar. But essentially, if we go back to the root, we see that there's something called NIDDK Pilot Funding News. So if I click on that, this is a special database that we created because we are working with the different NIDDK centers that is a database of essentially the pilot grant opportunity and feasibility study opportunities that they have available. So a lot of NIH grants, especially for centers, present these types of opportunities. They're small pilot grants, about $50,000, short duration. But there is no place that we know of that you can go and search for these. So we created them. We have the ones from all of the centers we mentioned, but we've also gone out and, for example, found metabolomic centers and others that offer this type of opportunity. And so this is a specialized source that you can search for to look for these very specialized opportunities. So let's go back to funding again. A second use case that we got that looks a little bit more deeply into the advanced features was an interesting question that we got from a researcher who said, I'd like to know what study sections are reviewing grants in my area. So of course, you can go to Google and they'll take you to an NIH listing of all the study sections that are available. But when we were talking to him, we said, you know, perhaps one of the best ways to sort of strategize about what study section you might want to recommend that your grant be targeted to when you're asked for that information is actually to go and look at all the work that's been funded recently in your area and look to see what study sections reviewed those grants. So the researcher was not aware that NIH Reporter could actually be used for this. So we went through this use case using dkNET. So over here under funding, we said that there are opportunities, but there's also funded grants. And so if we click on this, the source of these funded grants is NIH Reporter. I'm sure many of you are familiar with them. Again, I always prefer table view. But if you go into the table view, it gives you a list of all grants that were funded that have metabolomics somewhere in their description or their title. So again, you might want to ask a question that says, well, gee, you know, I'm interested to see what what institutes fund this type of research. And if I click on my word cloud, I see actually the number one is the National Cancer Institute. Again, not surprising, knowing budgets and things, but that in fact metabolomics research is scattered across many different institutes, and NIDDK is not even one of the major funders of at least that keyword. But if I want to know about the funding agency, even if I do, or if I want to know about the study section, even if I do show more columns, that information is not available through dkNET. And that's because in dkNET, we're indexing these very large, complicated databases. We make a choice about what we think the meatiest columns are, the ones that you would want to know about. But as I mentioned in the beginning, we always encourage you to go to the source database to look for more. So in this case, we happen to know that study sections are listed, but we went through a little exercise that said, well, what type of grants are you interested in? And he said, well, it's going to be an R01. So we said, okay, we can use some of the filters that we have available to just show us R01s. And he said, well, I'm also interested in NIDDK. So how would you go about searching for NIDDK? We do provide some of these facets on the left, but you also always have the opportunity to come in and just type in an in-column filter like this, so that I can search for kidney, because that is a word that would get us NIDDK. And that essentially brings us down to the list of grants that were actually funded by NIDDK. And of course, I'm going to be submitting this soon, so maybe the only thing I want to do is have those that were funded in 2017. So I can click 2017. So I've used NIDDK to kind of narrow things down, but once I do that, I can click on the actual link here, and it will take me to NIH Reporter, not to the face page, but to the actual record that I'm looking at. So we always take you out to the source database when we can, but we try to drop you into the place where you were searching. So this is the NIH Reporter Interface. Again, it requires some poking and exploration, but if I click on details, I can see when I scroll down that here's the study section. So it's the kidney, nutrition, obesity, and diabetes. And if you use some of the query tools that are available from NIH Reporter, you can actually return a list of grants that were all funded after being reviewed by this particular study section. So this is, again, a way that NIDDK helps connect you to the resources that are available to you, makes it easy for you to connect with them, but also opens a window to all of the other opportunities and things that they have available. So I'm going to skip down now to the end here. That was a little bit of an advanced search, and even though we have tried to make it a lot easier than it would be currently without something like dkNET, you can see that actually it's still a bit of work, and you do need to understand how dkNET works. You need to understand a little bit about, once you get into these resources, the type of information that they have available to you. But again, we have control of the interfaces now into these resources. So if there's particular type of information that you would like, or something that you think would be particularly useful, we generally have the capacity to make it available, and we'd like to hear from you. The other thing is that there's a lot of human resource at dkNET. Since we spend our days and nights looking across the resource landscape, making decisions about databases, just like a good reference librarian, we often can help you and connect you to the resources that you need. So you should always feel free to contact us. There's a little contact us button at the lower right-hand corner, and ask us the question, and we'll be happy to help because we want to save you time. But we really do hope that you will spend, in fact, some time learning how to use dkNET. It's a little different than Google. It's a little different than databases. It's kind of a hybrid. And to try to encourage people to poke around and spend time in dkNET, we've actually partnered with NERSA, the Nuclear Receptor Signaling Atlas, on a little search challenge where we encourage researchers to learn how to use dkNET to find data sets that NERSA is looking for, and to kind of compensate you for the time and make it a little bit more interesting. We've done this in the form of a challenge. So essentially, there's a task that's put out on the website. All of the details are there, but you can win prizes such as Amazon gift cards, and also sponsored. Someone has sponsored a challenge prize of $500, which will go to the one who finds the most data sets. So again, we encourage you to join, or if you run a lab, this is a great way for your students and postdocs to learn how to use dkNET, and they can win some prizes while doing so. So that is our dkNET challenge. So are there any questions about the search and display? I know I've gone through a lot, but again, I kind of wanted to show you how you can use dkNET and the information sources it has to address interesting questions. There are many more search features that you have available, but I've tried to only limit this to a subset and focus on the community resources. So Koei is not poking me, so I'm assuming again that there are no questions, but at any point feel free to interrupt. I wanted to very quickly go over this other aspect of dkNET that I talked about in the beginning, and that is dkNET and its support of rigor and reproducibility, and how it can actually help you a little bit with the new NIH guidelines for rigor and reproducibility, where all new grants are required to provide certain types of information as a appendix before the grants can be reviewed. So to do this, I want to step back a little bit and talk about something that was called the Resource Identification Initiative. So this initiative was launched back in 2014 through an organization called Force 11, the Future of Research Communications and eScholarship, also with a project that my colleagues and I work on. We come from the neuroscience community, called the Neuroscience Information Framework. And back in 2009, when we were trying to experiment with things like text mining to look at what resources were being used in the literature, because that was our job, to look for resources, we uncovered a rather disturbing phenomenon. And I think this is well known to almost everybody, but that was when you looked at the material and methods section of a paper, people would talk about things like antibodies, they would talk about things like plasmids or organisms, but they wouldn't provide enough information so that you could uniquely identify what antibody they were using or what organism they were using. And of course, we all know that this is of interest to us if we're trying to design an experiment, but more importantly, when we troubleshoot an experiment, these types of research resources tend to be key drivers of the variability that we see across experiments. So we knew that we had to introduce a new reporting standard for research resources. They were all over the place. Some of them provided vendor names and locations. Some of them provided URLs. Some of them provided catalog numbers. And essentially, the way that we chose to solve this, and this is well known to you that do genomics research, when you publish a paper with a specific gene, you provide a GenBank accession number. And that uniquely identifies the gene piece that you've used. So the the idea was, because we had these large aggregations of catalogs and registries for antibodies and organisms, that we could do the same for some of these research resources. So we launched the Resource Identification Initiative, asking authors to put in these RRIDs, and you see one of these below highlighted in orange, for key research resources. But we worked with a lot of different publishers and a lot of different journal editors across fields to say, we don't just want these identifiers in. There's a couple of conditions. We want them to be search friendly. We don't want to have to use Scopus or Web of Science or any citation index. We want to be able to type an RRID into Google, for example, or PubMed, and be able to return a list of papers that have used these research resources. We don't want to deal with 6,000 different citation styles. We want it all to be uniform across publishers, because again, the idea is that you should be able to identify what other papers were published using this research resource. And ideally, it should be available outside of the paywall. But we weren't entirely successful with number 3, although you can in fact find these through Google Scholar. But we were successful with 1 and 2. And from our modest pilot project, starting with 20 journals or 25 journals, we're now in over 250 different journals. And the Endocrine Society has just endorsed the standard for their journals, specifically for antibodies. So we already had been working in this area of resource identification. We've already gone through the use case of, again, someone says, I use an actin monoclonal antibody, and there may be 40 of these, and so you don't know which one they used. But I think the interesting thing about this is that this now lets us ask 2 very basic questions about the literature. One is, what resource did I use when I published my paper? So which exact actin antibody did I use? Which other studies, however, also used the same resource? And why is that important? It's important not only for us to be able to aggregate information or troubleshoot, but those of you who have been following the reproducibility crisis in quotes that's been affecting biomedicine know that it's been documented that oftentimes reagents, which are found to be non-specific or contaminated or discredited in one domain, continue to be used for many, many years afterwards, and people publish on them without being aware that there's information that might cause you to be concerned about the results. And so what's interesting is that RRIDs are currently in just about 3,000 papers, but we're already starting to see people cite resources where problems have been identified. And so essentially these RRIDs become an information channel over which we can layer information about how they perform. So here, for example, is a cell line, CVCL0027 is its accession number. And when we pull information from Cellosaurus about the cell line, we see that it's problematic. There's a problematic cell line that's been misidentified. Now, in the case of this particular article, that may not affect the results of the studies, but there's been some very high-profile cases where people thought they were studying one type of cancer and they were studying another type of cancer, and where it very much did matter. So these RRIDs, although simple to do, are in fact extremely powerful, because they, again, allow us very easily to be able to essentially recall all papers, or at least put an erratum or a notice of all papers that used a particular resource. So we're really encouraging people to use them, and we appreciate the support of centers like HERN in supporting the RRID initiative. So in the new NIH guidelines in Rigor and Reproducibility, this actually turns out authentication of reagents turns out to be one of the key features of their new guidelines. And so dkNET has provided a section on the website where we present the Rigor and Reproducibility guidelines. We've aggregated information that has come from different libraries, different recommendations about how researchers should go about addressing this. Here, for example, are the 4 focus areas that they're looking at, but in specifically the area of authentication of key biological and chemical resources. Here's where we think that dkNET can really, really help you. We don't want to get to the point of publication before you find out that the cell line that you used was problematic, or that there's a specificity concern in the antibody. So we've launched a beta service where essentially One, you want to properly identify your resource, so you'd want to include RRIDs in your grant application. We give you the metadata standard, which says you shouldn't just put in the RRID, but you should put all this other information in. But then we also provide a reproducibility report. And so in April, we launched this, and essentially, if you go to dkNET, we give you a tool where you can enter in all of the resources that you are planning on using, and we will generate a report for you. So for example, here, we've introduced one of the cell lines that, here's a report that says, I want to use the cell line, and it says, oh, did you know over at Cellosaurus, they say that this has been discontinued. So we think that this is a very powerful tool whereby we can help people, researchers in dkNET, provide these appendices. Also, if there is validation techniques that have been used, we provide the source of information. So that is, I wanted to make sure that we left time for questions. Again, the Rigor and reproducibility authentication is in beta testing right now, and we certainly welcome you to try it and give us feedback about whether you find out the section is actually useful. But again, I want to encourage everybody to engage with dkNET, engage with us. We know that there are things we can do better. As we said, this is a lot of information, and we're experimenting with ways to make it easier to search and to use that information. So we have surveys, we have various feedback mechanisms. If you ever have a suggestion, or just if you get confused and lost, we would appreciate you letting us know. If you know of important tools and databases that we don't have, and you want to recommend them to us, you should let us know. Again, this is a resource for you and your fellow researchers, and both basic and clinical. And so anything we can do to make it easier for others to find information is appreciated. I went through some basic search functions that are available, but if you sign up for an account, there are actually many more things you can do. So you can sign up for alerts, you can save your searches and rerun them at different times. There's a lot of different features that are available. So we encourage you to sign up for your dkNET account. We also have mailing lists and other things that can be used to keep you informed. And if you're a Twitter user, we have dkNET, and you can follow us on dkNET. And again, put in another plug for the dkNET NERSA Challenge. It may not be in your area of expertise, but again, it's a reason to sit down and engage with dkNET, to learn how it works, to sort of poke around in the different databases that we have. Think about clever ways that one can perform searches. A lot of times, as you saw with some of the searches, you may think, for example, that when you're looking for funding opportunities, that you should look only in the funding opportunities database, but then you say, hey, maybe I can find out which institutes actually fund this, and maybe I can look for more targeted things that way. So there's often very clever ways of using data and data sources. So I think that that's about it for my formal presentation. Again, we have some time. I wanted to make sure that we had time so that you can play along, you can ask us questions, you can give us your own use cases. We'd be happy to sort of work with you. And that's that. So we're going to unmute everybody. So if you'd like to talk, that way you don't have to type.
Marianne, for the previous slide where you had this beta, rigor, and reproducibility feature, does it also identify all the other individuals that would be or that may be already using that particular resource?
So I believe that it does give you a report of all the papers that were published with it. If it doesn't right now, it could, because that's the beauty of RRIDs, is that it actually lets you very easily say, here's all the other papers that were published with it.
Right. That's not, I mean, not all resources will be in the public domain, but if they are, that's really a nice feature.
Yes, it is. And that's actually a very good suggestion, because I think right now we don't have that because it's available elsewhere. But I think it's a really important backup to say this antibody has been used across all these domains.
It's not here.
And that gives you, again, some, you know, some valuable information. So good suggestion. Any other questions? No. Joey says no. Okay. Well, if there are no other questions, again, we are here to help. This little report an issue thing down here where we just said, I just put in a ticket to say, change that, and change that to say, this is really the help center. So if you can't find what you're looking for, or if it's difficult, we're always happy to assist you and use the knowledge that we have to maybe, suggest some strategies. So I think the human element in dkNET is the most important element, and we really encourage you to let us know, how we're doing, but also how we can help.